Testing. 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 All right. We're good to go. We are live. Welcome to another Hoopo stream. Check, check, testing, testing. Tsetse fly. Tsetse fly. We got two papers for you today. We got, so I was checking out papers for the rest of this week, and I found two different papers, both using diffusion models with fMRI images. So we got brain diffusion for visual exploration, cortical discovery using large-scale generative models. So this is coming from Carnegie Mellon, and this was released just Monday, I think was the 5th. And then we got another paper. This one's out of kind of a larger collection of different groups here. You got everybody from Eleuther AI and Stability AI, but also Medical AI Research Center in Princeton, École Normale Supérieure from PSL. I don't actually know where that is. University of Toronto. Maybe this is somewhere in Quebec. But I thought it was too much of a coincidence to not kind of potentially look at both papers. So I'm going to try to do something a little bit new today. I'm going to try to basically kind of bounce back between these papers and see if we can find a common thread or learn a little bit more by kind of comparing and contrasting the different things that they do. So let's go ahead and get started by just reading the abstracts. All right. So check. I study neuroimaging. I'm pumped for this. Pretty cool. Thanks, Drac. Maybe you can help us answer questions. I do not know very much about fMRIs at all, so I think it's functional magnetic resonance imaging, right? Is that what the F in fMRI stands for? Yeah, functional. Okay. A long-standing goal has been to elucidate the functional organization of the brain. Right, so one of the biggest problems, I think, in brain science right now is that it just kind of looks like one big mass, and we have to figure out, okay, well, what are individual parts of this brain doing? And I think we have some idea of like what the different parts of the brain do, but we certainly don't understand it entirely, right? I think there's still a lot of work to be done in terms of what is happening at different parts of the brain. Within higher visual cortex, functional accounts have remained relatively coarse. So I think the visual cortex is the part of your brain that processes the information that comes from your eyes. Specifically, I think it's in the back of your head. Visual cortex. Yeah, it's like this part here in the back of your head, which is kind of cool because it basically means that whenever you look at something, it goes straight through your entire head. It goes basically into your eye, through your optical nerve, and then right to the back of your head. So that's always kind of cool to think about. Functional accounts remain relatively coarse, focusing on regions of interest and taking the form of selectivity for broad categories such as faces, places, bodies, food, or words. Okay, so people have been able to identify regions that are associated with these kind of coarse concepts. Because the identification of such ROIs has typically relied on manually assembled stimulus sets. Okay, so probably data sets that people have gathered, and then they show you this data set, and then it's things like faces, places, bodies, food, and then they see where it lights up. Consisting of isolated objects in non-ecological contexts. Okay, so this is just a fancy way of saying 2D images that are very limited. Exploring functional organization without robust a priori hypothesis. So this is literally the Latin of beforehand, prior. So whenever we talk about statistical priors, it's like that's literally the same root word, and it all comes from Latin. hypothesis has been challenging. To overcome these limitations, we introduce a data-driven approach in which we synthesize images predicted to activate a given brain region using paired natural images and fMRI recordings. Okay, so image synthesis based on fMRI recordings, aka fMRI to image, bypassing the need for category-specific stimuli. Our approach, which they call brain diffusion for visual exploration, brain dive. I like it. I like the name. Builds on recent generative methods by combining large-scale diffusion models with brain-guided image synthesis. Validating our method, we demonstrate the ability to synthesize preferred images with appropriate semantic specificity. So you actually want to be able to generate what you're saying you're going to generate. For well-categorized, category-selective regions of interest. We then show that BrainDive can categorize differences between ROIs selected for the same high-level category. Finally, we identify novel functional subdivisions within these ROIs. Okay, so maybe we're getting a finer level of resolution, right? They're talking about how right now brain regions of interest are relatively coarse, but then maybe through this work they can find even finer subdivisions of these ROIs. These results advance our understanding of the fine-grained functional organization of the human visual cortex and provide well-specified constraints for further examination of cortical organization using hypothesis-driven methods. All right. Time series, first thought you should have with fMRI is task-oriented or not. Yeah, so the brain is a process, right? It changes over time. So I agree with what you're saying that to me, this is almost kind of like the difference between images and video, where you can do classification on images, right? You could probably do classification on like frozen snapshots of the fMRI activation. I don't know what the right word there is, but then you could probably also do classification on like a sequence of fMRI snapshots, right? The same way that you can do video classification. And those two things are very different. So maybe there's an analogous thing here. All right, let's look at the abstract for this second paper, see how much it rhymes with this first one. We present MindEye, a novel fMRI to image approach to retrieve and reconstruct viewed images from brain activity. Okay, retrieve and reconstruct is a little bit different than here. They use generative AI, but I think they're actually referring to the same thing. They just chose a different phrase. Our model comprises of two parallel submodules that are specialized for retrieval using contrastive learning. Okay, so contrastive learning is the technique behind CLIP, right? Contrastive language image pre-training. And contrastive learning, all it means is that you're comparing and contrasting two different things. So in the word of clip, or in the land of clip, what that means is you're basically saying this piece of text and this piece of and this image should be the same embedding point in some large manifold that we don't know yet, and then you pull the things that are similar together, and then you push the things that are different apart, and in that way you kind of shape this embedding space, and eventually that embedding space can be used to retrieve similar images given an image, or retrieve images given a text, and so on. So immediately I think that what they're probably going to do in this paper is they're probably just going to figure out a way that actually they tell you right here, can map fMRI brain activity to any high-dimensional multimodal space like the clip image space. So they're basically going to create some kind of encoder that can map whatever the raw fMRI data type is into a vector, and that vector represents a point in a multidimensional latent space or a multidimensional embedding space, such as the clip space. And then once you do that, you should be able to find nearest neighbors around that, and that'll be supposedly images that are similar. Enabling images reconstruction using generative models that accept embeddings from this latent space. Yeah, so then once you have this latent vector, right, this embedding that says this is what my fMRI is telling me a point in this clip image space, well, then I can use that point in the clip image space to condition a generative model, like a diffusion model, to generate an image. We comprehensively compare our approach with other existing methods using qualitative side-by-side comparisons, quantitative evaluations, ensure that MindEye achieves state-of-the-art performance in both reconstruction and retrieval tasks. I don't know how competitive the state-of-the-art is for fMRI imaging. You know, like, obviously they didn't compare to this paper because this paper came out roughly at the same time. So I don't know if this is necessarily impressive. It might just be the case that state-of-the-art is actually quite easy to beat in this particular niche. MindEye can retrieve the exact original image, even among highly similar candidates, indicating that its brain embeddings retain fine-grained image-specific information. Can retrieve the exact original image. It kind of sounds like they're training on or testing on the training set. You know what I'm saying? Like, what do you... Okay, we'll be curious to see how they separate the data that they use to train this brain embedding space versus the data that they use to test it. This allows us to accurately retrieve images even from large-scale databases like Leon 5B. So this is a data set that we saw people using. I think Stable Diffusion was actually trained on that, to be honest, but it's a very common image data set. We demonstrate through ablations. Let me actually highlight it in the red, which is usually our data set color. We demonstrate through ablations that MindEye's performance improvements over previous methods result from specialized submodules for retrieval and reconstruction, improved training techniques, and training models with orders of magnitude more parameters. Easy way to get state-of-the-art is just train a bigger model than pretty much anybody else did. Furthermore, we show that MindEye can better preserve low-level image features in the reconstructions by using image-to-image, which outputs from a separate encoder. Image-to-image. Low-level image features. So low-level image features here basically means usually things like texture, you know. Low-level represents the fact that most vision models like a VIT have basically this notion of hierarchy, right, where you have a lower level and a higher level. The higher level represents kind of more abstract semantic concepts, and then the lower level represents more things like texture, you know, like actual kind of like edges and stuff like that. All code is available on GitHub. Okay, so yeah, both of these papers are largely doing the same exact thing, right? They're basically going from fMRI into image space. This one is obviously going to be using Clip, right? They're basically going to be leveraging the fact that Clip has a very, very good image embedding space, right? That has been shaped with language as well. That's kind of the secret superpower of Clip, is that the embedding space that it creates and you can project images to has been shaped over time with the semantics of language, right? So that's what makes the Clip one very powerful, right? It's leveraging the fact that language is a very good prior. So they don't mention Clip at all in this abstract, but we'll see exactly what they do. Okay. I don't think we're going to have time to read everything here, but like, let's see if we can kind of maybe get a little bit of skimming done here. Task-oriented refers to the fact that the subjects were shown images instead of just letting them idle. Can equals resting state fMRI. Doesn't necessarily have to. Okay. So there's some kind of difference, Drac, is what you're saying. That's whether or not you're sitting there and just like kind of like not being shown anything versus having images flash. I don't know, you flash images every like second. I don't know like how fast the task is. Yeah, absolutely. The problem of decoding environmental inputs is fundamental to the Reese field of neuroscience. Improved computational approaches. Neuroimaging methodology is the domain is functional magnetic resonance imaging, where neural activity is measured by detecting changes in blood oxygenation. Okay, so largely what's happening is you're basically determining how much oxygen there is in the blood in all the different vessels in your brain, and that's being used as a proxy for activity. I think that's a pretty good proxy, but it's important to note that it's not actually looking at the neuron activation, right? The neurons in your brain, the way that they fire is they basically fire using electrical activity and they kind of spike. So this isn't measuring actual neural activity in terms of like the electrical spikes. This is just measuring it or using a proxy of blood oxygenation. fMRI decoding is already being used in real-time clinical domains and has potential for novel mind-reading applications in brain-computer interfaces. Previous works mapped fMRI to the embeddings of an image generation model via relatively simple mapping, usually ridge regression. So people have been doing this before. That's actually kind of interesting. If we actually look at these, how old are these? So we have a 2021 paper, we have a 2022 paper, and then we have a 2023 paper. Okay, so that's not super long, to be honest. It seems like it's actually relatively recent that we've been able to that people have tried to map fMRI data points into some kind of image embedding space. We propose Mind's Eye, a novel approach via mapping large-scale multi-layer perceptrons. So a multi-layer perceptron is a fully connected neural net that has multiple layers, right? Just think of a neural net in your head. And large-scale doesn't necessarily have a specific meaning, probably just means that they have a very wide and very deep net. Contrastive learning and diffusion models to achieve state-of-the-art image reconstruction. See Figure 1 for select samples. Okay, so here are some pictures. So we got the image that they saw. We got the reconstructed image. We got the image that they got, the reconstructed image. This is a weird way to lay it out, but basically they see this bear and then the brain of the person is measured. Then they take that brain measurement, they embed it into the clip latent space, and then they say what other images are close to that in the clip latent space. Or actually, that's not what they're doing. What they're doing is that would be retrieval, but here they're doing reconstruction, which is they then condition a diffusion model on this particular point in the clip embedding space, and you end up getting this, which is not just matching the semantic context here of like the bear in the grass, but it's even more accurate than that. It's like the exact size of the bear relative to the picture, and then also the orientation of the bear relative to the picture. Obviously, these are going to be cherry-picked, so take this with a grain of salt. These are probably the best results that they have. So, I don't know. We'll see exactly if they show us some results that are a little bit less good looking. All right, let's look at the kind of next part here on this first paper. DRAC. You basically get task positive and task negative signal, and then you see what lights up uniquely to the region whose activation is associated with the task. Okay, so basically there's always activation happening, and then whenever you have the specific task occurring, you record that, and then you basically just do the difference between the quote-unquote like resting state and then the task state. So it's like always a difference between an already previously existing brain state. I guess that makes sense. Previous studies have provided important evidence that regions in the higher visual cortex preferentially process complex semantic categories. These important discoveries have been primarily achieved through the use of researcher-crafted stimuli. So this paper just seems to really talk about this manually assembled stimulus sets, researcher-crafted stimuli. Like it's really kind of like drilling deep this idea that the stimulus that's being used to kind of elicit this brain activity is researcher-crafted. I don't think that necessarily is that bad, Like I feel like what else are you going to do? Hand-selected synthetic stimuli may bias the results or may not accurately capture the complexity and variability of natural scenes. Okay. Sometimes leading to debates about interpretation and validity of identified functional regions. Mapping selectivity based on response to a fixed set of stimuli is necessarily limited, and that can only identify selectivity for the stimulus properties that are sampled. Okay. Introduce a generative approach for synthesizing images. Several recent studies have yielded intriguing results. And then here we go. Images generated. So I guess the top part. Okay, so these are also generated, but we don't know what the original image is. Where is the original image? We find that brain signals, blah, 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 strengthening the evidence. Okay, so we don't know what the image that they showed the person is. As far as I understand, these are basically just images that are generated. So, I mean, all they're showing you here is that stable diffusion conditioned on a embedding is able to generate images. Further leverage state-of-the-art diffusion models in identifying fine-grained functional specialization in an objective and data-driven manner. Data-driven manner, like, this is just like, anytime someone says data-driven, I just like, everything is data-driven. You know, every single machine learning approach is data-driven, if you want to think of it that way. So I don't think that doesn't sound as cool as they think it sounds. We replace these prompts with maximization of voxels in given brain area. So voxels is a little tiny square inside a volume of space, right? Whenever you take a larger volume of space, you break it up into these little voxels. If you've watched any of our streams where we look at nerfs or any kind of like 3D stuff, voxels are a big part of that. And it seems like basically when you're doing this fMRI, you're also fundamentally deciding on some resolution of 3D space, and that resolution is going to determine the size of your voxels. The result being that resultant synthesized images are tailored to targeted regions and higher order visual areas. Because the synthesized images are optimized to maximize the response of a given subregion, these images emphasize and isolate critical feature preferences beyond what was present. We further performed several human behavioral studies to confirm that the semantic identities of our synthesized images. We established that BrainDive can synthesize novel images for category-selected brain regions with high semantic specificity. So what does that mean? Is that just dog versus cat, or are we talking about like person facing to the left versus person facing to the right? You know, semantic specificity just means that the concept is increasingly more specific. We further show that BrainDive can identify ROI-wise differences in selectivity that map to ecologically relevant properties. ROI stands for region of interest. Building on this result, we are able to identify novel functional distinctions within subregions of existing ROIs. Such results demonstrate that BrainDive can be used in a data-driven manner to enable new insights into the fine-grained functional organization of the human visual cortex. So they kind of mentioned, this is now the second time that they mentioned this subregions of existing ROI. So it seems like this paper is more trying to talk about how they're going to be able to identify even smaller subregions, even smaller voxels within the brain that are maybe not just for body, but specifically for left leg or right leg or kind of concepts like that, more specific semantic concepts. But I don't know, to me, I feel like that's also just a result of the resolution of whatever you're using to get this fMRI images, right? If you had a better machine that could give you 10 times the amount of voxels, I feel like you would get a much better result in terms of finding subregions of existing ROIs. So I don't think that the subregions of existing ROIs are the result of just poor planning. I think it's just because people probably were using older fMRIs. So the voxels are just coarse. Map flattened spatial patterns of fMRI activity across voxels. Flattened here, so they're probably going to be taking the 3D information, which is in these voxels, and then just flatten it out into a sequence so they can feed it into a transformer, if I had to guess. Or no, it's not even a transformer. It's an MLP. That's sloppy. They should have definitely had a transformer there. The retrieval is contrastively trained and produces disjointed clip MRI embeddings. Okay. So for retrieval, they basically just freeze the image model probably and then just kind of train this MLP backbone for embedding the fMRI that have high cosine similarity. Cosine similarity is how you determine whether or not 2 points in this clip embedding space are similar, right? Those 2 points are basically just a vector of numbers, and when you do cosine similarity, you're basically looking at what is the cosine between these two vectors, and cosine similarity, right? Like anytime you see that, just think about this, right? Where if you have one vector, this is a 2-dimensional space. Obviously, the embedding space of clip is probably thousands of dimensions, but you fundamentally have 2 vectors and you have some angle between them. The smaller that angle is, the more related those concepts are going to be, right? There you go. Cosine similarity. The similarity between vector A, which here represents apples and oranges, is A dot B, so there's a dot product, and then divided by the magnitude of A over B, and that's going to give you a single number, and if that number is small, these two things are similar. If that number is big, these two things are not similar. Oh shit, Drax going crazy over here. If you let someone just chill and think about nothing, then measure the signal, and then take the first PC and PC2, I think your PC, you probably mean principal components. Principal component analysis is a dimensionality reduction technique. You get the default mode network and executive function network. I know about the default mode network. The distinction with data-driven is in contrast to hypothesis-driven research. It's not pseudo-profound. It's a fundamentally different way of approaching an experimental design. Okay, so data-driven is, let's look at the data and see what happens. Hypothesis-driven is, I have this idea, and then can we figure out if this idea is correct? So I see what you're saying. Specifying ROIs is generally regarded as almost fully departed from the data-driven approach. Okay, so if you already come in with some prior about here's the specific regions and here's what each region corresponds to, that prior is almost like a hypothesis. So that's kind of what you're talking about, Drac, there, is that hypothesis-driven, you come in with the prior of I already know what these regions are, versus data-driven, it's, okay, I'm going to have no assumptions, let's just see what ends up happening and which things activate according to which semantic concept. As for voxel counts, you get to sub-millimeter voxel sizes. That's pretty cool. I didn't realize that the voxel size for MRIs are sub-millimeter. That's pretty impressive. Anything else is artificially inflated by simply subdividing the voxels or meshes when the vertex edges. This really great parsing for out of field. I'm actually in awe, for real. I appreciate it. To reconstruct images, blah, 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 we train a diffusion prior to taking the outputs from the MLP backbone and produce aligned embeddings suitable as inputs to any pre-trained image generation model that accepts clip image embedding. So this is basically going to be a stable diffusion. We train a diffusion prior to take outputs and produce aligned embeddings. In order to ensure that our reconstructions also match the original image's low-level features, we train a separate encoder that directly maps voxels to the embedding space of Stable Diffusion's variational autoencoder. Obtaining blurry image reconstructions that lack high-level semantic content but perform state-of-the-art on low-level image metrics. So I don't know whether here they're trying to refer to the fact that they're doing the diffusion in a latent space. So one thing that we've seen over and over again with these generative diffusion models is that a lot of times people will perform the diffusion in the latent space, not in the actual image space. So it kind of sounds like they're saying that they train a separate encoder that goes from the voxels, which I guess is the raw fMRI data, into this latent space of the VAE, right? Variational autoencoder is best picture. VAE. Let's see if we can find a good picture. Yeah, this is kind of a good picture. But basically, you always see these two channels that go like from here to here, right? You have your original data, you have an encoder, you have this bottleneck. The reason it's called a bottleneck is because you could think of it like the width of the neural net is choked there, right? Like there is a lot more numbers here in the input than there is here. The dimensionality of the input and the dimensionality of the bottleneck is way lower, which means that you're forcing this encoder to learn a way of compressing the information. And then you take that bottleneck, that little vector there, that little latent embedding, whatever you want to call it, and you feed that into a decoder, and the decoder should reconstruct the original image. And because you know exactly what the original image is, now you can actually very easily just use this as a kind of like a self-supervised objective, and then push gradients into this, and then over time what you eventually end up getting is you end up getting a encoder and a decoder, and really they should be as similar as possible. And there's different things that people have done. So for example, this is the VQ-VAE. It's a quantized VAE, and in this case what they're doing is that little latent space here, right? That little bottleneck vector, that little Z vector, they're actually quantizing it, which means that they're limiting it to a specific code book of possible values. So that's one way that people have one extra thing that people have done, but there's all kinds of variants of these. And in this paper, they're using a variational autoencoder that Stable Diffusion has, I guess. Obtaining blurry image reconstructions that lack high-level semantic content but perform state-of-the-art on low-level image metrics. Lack high-level semantic content, blurry reconstructions. Combining the high-level semantic pipeline with the low-level perceptual pipeline setting allows mind's eye to output state-of-the-art reconstructions across both low and high-level image metrics. So low-level image metrics, like when somebody says that to me, I'm thinking like the mean and variance of like the red channel, or the mean and variance of the blue channel and the green channel, like, or potentially the, because it's not features here. They're saying metrics. So what is the metric that they're using to evaluate whether or not the low level stuff is okay or the high level stuff is okay? So we're gonna need them to kind of specify that a little bit more, because right now it's not entirely clear what they're doing here. In addition to image reconstruction metrics, our disjointed clip fMRI embeddings attain state-of-the-art performance on image retrieval and brain retrieval metrics. Okay, they keep mentioning state-of-the-art, but again, I don't know how many other papers they can even compare to, so it might just be the case that they have nothing to compare to. Image retrieval refers to finding the original scene image out of a pool of other images, given the brain sample. Right, this is what they're doing with their step 1 here, where they basically train a MLP backbone to project fMRI into a clip embedding space, and then they just use that clip embedding space for retrieval using cosine similarity. Brain retrieval refers to finding the brain sample given an image. Okay, so actually now you could do it the opposite way too. So if you have this encoder that projects from fMRI into clip, you could actually go the opposite way as well, as long as you have a decoder. Mind's eye finds exact top 1 matches in the pool out of NSD test samples with 90% accuracy for both image and brain retrieval, outperforming previous state-of-the-art, which showed less than 50% retrieval accuracy. So how old is this state-of-the-art? Ooh, that's actually quite recent, 2022. Okay, so they're beating the state-of-the-art, but it's not like they're comparing to some 1980s paper. They're comparing to 2022. I don't even know if people were doing this in the 1980s, though. Mind video is the main comparison I would make, released 2 weeks ago. Okay. Our main findings are specialized submodules for retrieval using contrastive learning and reconstruction. Low-level features are color, size, position. Those are very well mapped, or those are already very well mapped in the human visual cortex. So we already kind of roughly know if there's something here in the corner of your eye versus in the bottom of your eye, what part of the brain is going to activate for that. I guess that kind of makes sense, right? Because in your eye, you have the cones, right? The cells that actually receive the light, right? And then there's kind of a mapping, right, between every single one of those cone cells or rod cells or something like that, right? And then a specific part of your brain. So, right? And the CNN, human visual cortex. But I've heard a lot of people say that the actual design for the ConvNet was based on the way that the visual cortex is organized. So there is kind of like a, yeah, maybe not this. Where is it? Here you have the cell, little neuron, passing a signal to another little neuron. This seems like a kind of cool article. Maybe we read this one. But whenever low-level features are mentioned, this is what I call low-level features here, right? Low-level features, mid-level features, high-level features. So high-level features, you're almost seeing like specific patterns, eyes, specific objects, you know, like a rear view mirror, something like that. Low-level features, these almost look like edge detectors, right? So I guess this is maybe what your brain looks like. Your brain has some kind of encoding and decoding kind of process going on. You have these different regions. I guess V1 is the first one, and then V2, and then V4. So V2 and V4 are kind of the higher level equivalent. So V2 and V4, I guess, should have more semantically meaningful high-level features, and then V1 should be the low-level features, such as this. The problem is that this is low-level features and high-level features for a ConvNet, right? But in a transformer, it's not going to be the same thing, and I think in your brain, it might not even be the same thing. So this intuition isn't necessarily going to map IMO. That part of the actual physical space on your visual cortex. Hupo's live deep dives into these ML papers are some of my favorite content in the space. I appreciate it, Jonathan. Look for radial projection images. That's what visual cortex signal is actually represented at. Radial projection images. Oh, shit. Yeah, so one actually cool idea that I remember someone explaining to me at one point is that when you look at ConvNets now, they're in this grid, right? There's this kind of like the way that pretty much everything is designed around images, which are basically a two-dimensional array of pixel values. But a much more natural way of representing would be a circle, right? And I remember someone explaining to me an idea where basically rather than having a convolution where you have a kernel that is basically a 3x3 grid, you have a convolution where the kernel is a circle. And you basically sample maybe like 10 points around a circle and then 10 points around a bigger circle, you know? Where basically think about the kernel being like a circle and it like moves in a circle, right? So every rather than everything being square, everything is a circle. I don't think it worked very well, but it's a very cool idea, right? Where we have this kind of prior of this square grid, and that prior just is the result of just the way that our computers work and the way that our computers are discretized and the way that images are kind of represented in our binary computers. But in the real life, there's probably more of this kind of radial symmetry as opposed to this kind of grid type pattern. Okay, let's get back to this. Oh no, okay. Specialized submodules for retrieval enable a single model to achieve state-of-the-art results across both tasks. Mapping to a deep MLP with a parameter count. I really wish they would have done a transformer. That just seems so much cooler. Anytime I see MLP, I just like feel like they left something on the table. With parameter counts order of magnitude higher than previous methods, does not produce overfitting. So obviously, the bigger your neural net, the bigger your model capacity, the more you can overfit. This is especially dangerous if you have a very small data set. So if you have a very big neural net and a very small data set, you're basically guaranteed to overfit. So a lot of times whenever people are doing these kind of more kind of science-y kind of experiments and they have smaller data sets, they'll generally kind of use a very small multi-layer perceptron. So I guess here they're kind of boasting that they don't have overfitting despite the fact that they have a huge MLP. A novel bidirectional version of mix-up contrastive data augmentation. Okay, so data augmentation is a technique that is pretty standard where basically you corrupt your data as you feed it into the model. I'll augmentations. This is a very popular library. Augmentations, GitHub. Albumentations, that's what it's called. There's a couple different ones, but basically they're libraries that will basically fuck with your image before you feed it into a neural net, and that there's a specific one called cut mix that they're referring to here. Where is it? They call it mix up, but mix up I think refers to one where you kind of basically crop and then move it. Let's see if we can actually get a picture of a mix up augmentation. Yeah, here we go. Where's the mix up? Show me a pic. These pics suck. Let's get a better pic. Mix up. There you go. So a mix up, you have 30% dog, 70% cat, and then ideally you should predict that as 30% dog, 70% cat. So maybe this works well for low-level features, but I could see how this kind of confuses high-level stuff. I don't know how they're going to be doing that for fMRIs. That seems kind of intense. Further improves model performance. So data augmentation is a very common way to prevent overfitting because you're effectively inflating your dataset distribution. You're making your data bigger than it is by using data augmentation. State-of-the-art reconstructions for low-level image metrics can be obtained by independently mapping to Stable Diffusion's VAE latent space. If you map to the latent space of a VAE, I would call that an embedding vector. I wouldn't call that a low-level image metrics. I don't know. I'm still confused about how they're using this VAE here, but I think they'll probably explain it. fMRI to image retrieval can find the exact original image even among highly similar candidates, suggesting that fine-grained image-specific information is contained in brain embeddings, thus allowing retrieval to be scaled up to large datasets like Leon 5B to output images without generative models. Okay. So this is kind of interesting. What they're suggesting here is that, okay, you could take your brain embedding, the fMRI image or the fMRI data point, convert it into a vector, and use that vector to condition a generative model to generate an image, right? But they're saying that if you have a large enough data set, like Leon 5B, retrieval is generation. Generation is retrieval, right? If you have every single image in the internet, and you can think of a picture of a bear facing to the left, there is plenty of pictures of bears facing to the left that you can retrieve from the database, right? So this idea of retrieval is generation, if the database is large enough, that's kind of a powerful concept. Okay, so here we have the actual schematic. Let's see if we can pull the schematic for the other one here. All right, there we go. Let's compare these end figure to figure, dude. Let's do a figure off, a figure battle. So first step of the figure battle is, I think this one actually looked cooler. I like how much more simple this one is. I like the color and the text. This one looks a little bit grosser. I don't like the color. I don't like the text. I don't like the just raw amount of arrows. Like, what the fuck is this arrow? That's a little bit confusing. Okay, so just visual appealingness. I love this picture. The brain. This brain seems a lot cooler than this brain, so we'll give this one the edge on the brain as well. All right, but memes aside, let's actually see what they're talking about. Look for projection images. Gotcha. The semantic part throws me off. Maybe it should still be. I'm just saying the physical space that the retinal signal is projected to on the brain is a circle, and the neural signal is usually represented as that too. Okay. The reshaping mechanism learned by MLPs is not data dependent, while the one for transformers is. How do you feel about this statement? Reshaping mechanism. I don't know what you mean by reshaping mechanism, but a multi-layer perceptron is just routing things, right? At the end of the day, what a neural net is doing is it's basically creating a specific path, right? So like this neuron might learn to pay attention to this neuron, which learns to pay attention to this neuron and this neuron, right? And that's kind of what's happening in a transformer as well. The transformer, all it's doing is just basically replacing this kind of routing that is done through kind of explicit edges and nodes on a graph with this attention map. But it's kind of the same thing. You're letting everything in the previous layer access everything in the layer before, right? It's like full connectivity layer to layer, right? So a multi-layer perceptron, every single neuron can pay attention to any of the previous neurons, right? There's an edge between every single one of those, and that's similar to what happens in the transformer, right? Transformer neural net. In the transformer, it's the same thing. You basically, any part of the sequence can pay attention to any part of the sequence. Let me see if I can find a good pick. I don't know if I can find a good pick. Okay, I can't find a good pick. But I don't know what the reshaping mechanism is. Reshaping mechanism. That's just like such a broad term, like it can literally mean everything. Like it seems like we're running into like weird patents for whatever the fuck this is, like some kind of like industrial metal manufacturing. Okay, back to these figures. Let me actually take a big sip of this yerba mate here before I continue. All right, so we have start to end. We have a diffusion process. Obviously, diffusion processes have time steps, right? You diffuse over a series of time steps. 1, you have a diffusion model trained to synthesize natural images by iterative denoising. We utilize a pre-trained latent diffusion model, so they are diffusing in the latent space. You see here the xt and then xt minus 1. This refers to the latent at time step t and then latent at time step t minus 1. There's probably on the order of 10 to 100 different time steps. Here they kind of show it like 10, but you see this ... here. That means that there's probably more steps that they're not showing you. An encoder trained to map from images to cortical activity. Okay, so you see here, this is the actual brain. This is the back of the brain, the visual cortex. This is the, I guess, I don't know what RSC, PPA are. These are probably just different regions of the brain. Those are mapped, right? I guess this data, I don't know what format this data has. The way that they've been saying it, it sounds like voxels. So basically you have a bunch of occupancy grids. So basically maybe a number for each voxel. Every voxel has like a float 64, and then that float 64 represents how much blood there is in that particular point in space, if I had to guess. And then you might have a 100 by 100 by 100 grid of voxels, so that gives you 100 cubed number of voxels. Maybe there's some better way to represent that. I don't know how sparse that data is. I don't know if the majority of those voxels are empty or whether the majority of those voxels are full. But that gets fed into an encoder, this brain encoder. This brain encoder is going to produce a vector, right? That vector, gradient flow. This is the red is how the training happens. But that vector is somehow decoder. Okay, wait up. I'm getting confused here. The brain encoder takes the image and produces a brain region target. But you know what the real brain region target is, so you compare the generated brain region target with the actual brain region target. The difference between those, some kind of loss. I don't know if you can use like an L2 loss for that. I don't know what the fuck that looks like, but some loss. Then you use that to push gradients. You're going to use those gradients to improve your brain encoder, and you're going to use those gradients to improve your latent decoder. And this is the latent decoder for the latent diffusion model. So this is the same decoder that's used to basically project the image into a latent space where the denoising actually happens. An encoder is trained to map from images to cortical activity. This makes it sound like it's not a latent. I would assume it would map from latent to cortical activity. Our framework can synthesize images that are predicted to activate any subset of voxels. The use of the word activate here makes me think that maybe the voxels are basically binary. They're either activated or not, so maybe it's 0 and 1. Maybe it's like an uint 8 rather than a float 64. Shown here are scene selective regions on the right hemisphere, so this is only half of the brain. We visualize every 4 steps the magnitude of the gradient with respect to the latent and the corresponding predicted x naught. So this looks like a saliency map. So if you guys have ever seen this, saliency maps, these are a common way of kind of exploring what the inside of a neural net, what's happening inside a neural net. And it's basically you're looking at the little neurons and you're seeing which ones are actually being activated, right? So if you were to do a saliency map for something like a fully connected neural net, it's not going to be super, you're not going to really see anything that's interesting, right? But like once you look into an image, right, then you actually know the coordinate and you can map it to the original image, and then you can actually see, okay, this part of the dog has high activation with whatever the fuck the original thing is. You can also do saliency maps with sequences. So saliency maps for transformers are also a thing. Saliency map sentences. There's like one specific thing that... Yeah. So for example, I've seen saliencies for like a sequence as well, right? Where you can see stronger influence versus weaker influence. So you can see which parts of a sentence are kind of resulting in the final output. Go back. We visualize 4 steps. So magnitude of the gradient is basically how the gradient is fundamentally telling your neural net, you need to change the value of this little weight over here. You need to change it, add a little bit or remove a little bit, right? You're performing gradient descent. You're basically changing the weights of all the little parameters in your neural net. So like what the gradient is really is ultimately, it's basically just a little delta. It's a little delta on the weight. It's basically saying add a little bit or remove a little bit. So the magnitude of that is telling you how much you want to add, how much you want to remove. So I guess the fact that it corresponds to the real world scene is good, but it's not. It's actually very complicated. You see here how their edges are very defined, right? I guess this scene is all about edges. You have very crisp, dark edges. But like, for example, what is this, right? Like, why is there this little dot here? Like, is that... There's nothing that really corresponds to that there. I don't know. I'm a little confused by this, to be honest. Let's see if we can get a better intuition here. Okay, so mind-eye overall schematic. A high-level semantic pipeline maps voxels to clip embeddings for image reconstruction. Okay, so here's the voxels from the fMRI. I guess there's 15,000 of those, or N of those. The MLP backbone is 257 by 768. Is that really the big MLP? This whole time they were talking about orders of magnitude higher? That's it? It's 257 by 768? 256 layers? Like, what is this? This is just the dimensionality of the embedding? What is that number? But this is the actual MLP that projects from fMRI voxel space into clip image embedding space. Right? Here you just have a clip VIT vision transformer with a patch size of 14, so 14 separate patches. And you can see here the little lock means that it's frozen, so they're not pushing gradients into this. This part is frozen. You can feed the image, you get the actual clip vector here. You're saying that this fMRI voxel data point should correspond to this image, so basically this vector here that comes out of the clip image embedding, and this vector here that comes out of the fMRI embedding or encoder or in this case, it's just an MLP. So MLP, you have a contrastive loss there. So these two things are getting pushed together. And maybe they do negative examples so that you can push as well as pull, right? So contrastive training. So you don't just want to push together things that are pull together things that are similar. You also want to push away things that are different. So they probably sample a couple different unrelated images and then also use those in the contrastive loss. You have an MSC loss here. The MSC loss is between clip fMRI here. So you have a diffusion model. You see here multiple steps of a diffusion model. This part here, the output of the fMRI MLP is being used to condition each step of the diffusion. That's what I'm kind of interpreting here by these arrows. Really, I would have made the arrow go into actually like in between, right? Because the conditioning is happening on generation. It's not going into this thing. Okay, you could also use this embedding vector that comes out of this fMRI MLP in order to retrieve from a large database, right? This is the whole thing that they were arguing, where retrieving from a large enough database is the same thing as generating with a diffusion model, because a diffusion model is basically just a compressed representation of a huge data set. Disjointed clip fMRI for retrieval. Aligned clip fMRI for reconstruction. So this vector here should be very similar to this vector here, which is why you just have a MSE, which is the mean squared error, right? That's just making these as similar as possible. And that goes into versatile diffusion. The fuck is versatile diffusion? Is this the VAE? Yeah, here you go. Here's the VAE. This is the VAE. So the VAE, taking an image here, encoding it into this latent, right? Where's our VAE? Right? This little Z vector here, you're encoding your input into some latent Z vector and then using that to go back to your input, and these two images should be the same, so kind of a similar idea here. Right, you're going from this image here, encoding it into this latent, which is n by 4 by 64 by 64. And then you go back through the stable diffusion decoder into the original image. So notice how all the different image encoders are frozen. Notice how there's a lock here for the image encoder, there's a lock here for the image decoder, there's a lock here for this large vision transformer that's part of clip, there's a lock here for this versatile diffusion. This is also a diffusion process. I think that's what that means. I think they're taking this image here and they're using diffusion in image space conditioned on this vector here. That's probably what's going on here. So I think this is diffusion in the space of images, and then this is diffusion in the latent space. So that might be why outputs from a diffusion prior fed through generative models like versatile diffusion or retrieval tasks. A low-level perceptual pipeline maps voxels to the variational autoencoder used by stable diffusion to obtain blurry reconstructions, which are then used as initialization for subsequent diffusion-based image generation. Yeah, okay, so this makes more sense. So I thought there was only one diffusion model here, but there's not. There's two diffusion models. There's a diffusion process happening here, and then there's a diffusion process happening here. So yeah, and also I don't like how they're calling this like high level versus low level. Like this is not. It's low level because your latent here is just not that great. You know what I'm saying? Like ideally, whenever you actually do a VAE like this decoder should reproduce the same exact image. And like, yes, it's blurry, but that's just blurry because it's not perfect. Ideally, you wouldn't have to do this extra step here where you perform additional diffusion, right? So this diffusion process here is just basically starting from this blurry image and then just making it more crispy, right? But realistically, you wouldn't necessarily need to do that. Realistically, this here should just produce the image that you want. So A little bit confusing, a little bit complicated in terms of this pipeline. Let's look up what versatile diffusion is. Versatile diffusion is a multi-flow, multimodal diffusion framework that supports image-to-text, image variation, text-to-image, text variation. Let's see. First unified multi-flow, multimodal diffusion framework. So how does this work? What the fuck is this? Area of rocks? What exactly is this doing? Let's see if we can get the paper here. It seems like this is a critical part of what they're doing. That's why I'm kind of spending a second here to actually read it and see what's going on. Demo result of our versatile diffusion. Recent advances in diffusion, blah, blah, blah. In this work, we expand a existing single-flow diffusion pipeline into a multitask, multimodal network dubbed versatile diffusion that handles multiple flows of text-to-image and variations in one unified model. Multiple flows. Okay, so that's similar. Multiple flows. There's multiple flows, I guess, happening here, where you have something coming here and then something coming here. Pipeline design instantiates a unified multi-flow diffusion framework consisting of shareable and swappable layer modules that enable the cross-modal generality. What? What is this? One diffusion step. Choices of data layers, choices of context layers. Choices. So you can choose the context. Is that what they're saying? Each color line represents a single flow that represents one supported task. The VAE decoder or encoders on the far left are used only in training and are replaced with Gaussian noise inputs during inference. So you have an image, you're encoding it, it's going into a ResNet. You have a piece of text, you're encoding it, it's going into another ResNet. You have some cross attention here between the features that come out of this image and the features that come out of this text. That cross attention is also somehow being given a clip image vector. Okay, I don't really fully understand this, but it seems to me like all they're doing is they're basically just conditioning these modules in the middle of a diffusion process on vectors. So it's like, it's just a fancy kind of way of conditioning, I guess, which is, I guess, what's going on here, right, is they're basically doing a diffusion process for this, but they're conditioning it on this clip fMRI here. Damn. That kind of sucks, you know. Read both of these figures, don't really understand either of them, so I guess that's just the way it goes. Background on diffusion models, we can probably skip that. Brain encoding model construction. This is a little bit more interesting. Let's see. A learned voxel-wise brain encoding model is a function m theta. Okay, so a model parameterized by some parameters theta that maps an image I. I is a image which images have a dimensionality of a height and a width with 3 channels RGB, so all real numbers in 3 by h by w, to a corresponding brain activation fMRI beta values represented as an n element vector b. So okay, so we finally know what the actual fMRI data looks like. So fMRI data is basically a giant vector of length n where each value in that vector can be anything, right? R is negative infinity to infinity. And you have your brain encoding model. All it's doing is it's basically taking an image and then producing a B, which is what we saw here, right? You take the image and you encode it into a vector which basically represents fMRI beta values, aka brain activations. Past work has identified later layers in neural networks as the best predictors of higher visual cortex, with clip-trained networks amongst the highest performing brain encoders. Yeah, and it's kind of interesting to think about that because I keep mentioning this over and over again, but like I think that the reason clip works well is because language is a very good prior if you want semantically ordered embeddings, right? Embeddings that kind of have some notion of semantic concepts inside of them. So your brain is probably a similar thing, right? Your brain is using language to constrain the way that your visual cortex is organized. Probably it's probably not language like English, but it's like some kind of like brain language, and there's similarities between actual human language and whatever language is inside your brain. I don't know, that doesn't make any sense, I'm sorry. We utilize two component designs from our encoders. The first component consists of a clip-trained image encoder which outputs a k-dimensional vector as the latent embeddings. Clip-trained image encoder which outputs a k-dimensional vector as the latent embeddings. I think that's this part here. So this is basically the clip encoder. The second component is a linear adaptation layer. W equals n by k. B, I guess, so linear adaptation layer just means one layer neural net, right? And the one layer is going to have weights and biases. So here all you see is weights, and then you have a bias, which maps Euclidean normalized image embeddings to brain activation. So image embeddings, aka a vector that is normalized, and normalized in Euclidean space, which is basically just X, Y, Z, and then you can keep, you don't need to necessarily be limited to 3 dimensions there. So this B, which is the n element vector that represents brain activation, is coming out of this model parameterized by theta, which consumes an image, and this model is just one weight matrix times the clip of the image, the magnitude of the clip of the image. So this is your normalization right here. This is your Euclidean normalized image embeddings, and then you're just adding this bias and multiplying by this weight. Optimal W star and B star are found by optimizing the mean squared error loss over images. So this is it's kind of cute, but this is basically just deep learning 101 right here, right? It's like you're optimizing a weight matrix and a bias matrix by pushing gradients that are coming from a mean squared error loss. So this is almost like the simplest form of deep learning, or of learning. It's not even deep learning because it's not even like multiple layers. You're talking about one layer here. We observe that the use of normalized clip embeddings improves the stability of gradient magnitudes with respect to the image. Yeah, so why would you want to normalize these? Because if you don't normalize this, some parts of this image embedding, which are basically a vector, some of them are going to be very big, some of them are going to be very small. So by normalizing them, you center it on 0 and you make sure that the variance is nice, narrow enough that you can fit it reasonably, so you don't have any kind of like wild swings back and forth. The DRAC, the similarities between how our brain works and how LLMs, both GPT and BERT, work are more obviously similar by the day. So what you're saying is totally sensical. Yeah, kind of a digression, but like I remember reading this stuff about how like technology changes the way that we think about things, and like actually whenever STEAM technology became a thing, like you had all these people and all these scientists that the way that they tried to interpret it, make sense of like human behaviors, was through the analogy of steam, right? Where they had this idea of like, oh, you have too much steam, and then because of that it leads to anger and all these problems. And it's like, we look at that now and we're like, dude, that doesn't make any fucking sense. Like there's no such thing as steam inside humans, you know, like what do you mean? But it's because that's the machines that they had. They had these steam engines, and they were trying to like make sense of everything in that same kind of mental model, that using that prior of the steam engine. And we're doing the same thing now, right? Where we're kind of familiar with neural nets and kind of diffusion models and transformers, so we want to think of everything as a neural net or some kind of machine learning model. But maybe if we project forward 100 years, they'll look back at us and think that, wow, they were stupid to think that the brain is a neural net, even though to us it seems like it's obviously a neural net. Brain-guided diffusion model. Okay, so finally we get to the actual part where they explain exactly what's going on here. Holy shit, what is this? This is a loss. All right, let's get to it. Brain dive seeks to generate images conditioned on maximizing brain activation in a given region. Conventional text conditioned diffusion models. The conditioning is done in one of two ways. The first approach modifies the function to further accept a conditioning vector. Yeah, so you can condition a diffusion process on a vector that can represent anything. You can condition it on a vector that represents a class. You can condition it on a vector, such as in this case, a clip vector. So here, right, the denoiser model, this is the denoiser, which is a function parameterized by parameters theta. It consumes a specific image at a time step or a latent at a time step, depending on whether or not your diffusion is happening in the latent space or in the image space. It's also consuming the time step itself, so some notion of how far along the process of diffusion you are, and then it's also consuming a context vector C. The second approach uses a contrastive trained image-to-concept encoder and seeks to maximize the similarity measure with respect to a text-to-concept encoder. Okay. Conditioning on activation of a brain region using the first approach presents difficulties. We do not know a priori the distribution of other non-targeted regions of the brain when a target region is maximized. They don't have a good prior for that. That's what they're saying. Overcoming this problem requires either to have a prior for this B. So B is the vector of fMRI values, and they say they don't have a prior for that, but I would say there's plenty of priors, right? It's probably a Gaussian prior could probably be good. Like, I don't know. You could probably look at already existing data and see if there's a prior there that you could assume. But the joint distribution of all voxels in the brain, to ignore the joint distribution that can result in catastrophic effects, or use a handcrafted prior that may be incorrect. Yeah, so they don't want to. This is, I guess, the point that you were saying, Drac, about hypothesis-driven research versus data-driven research, is they don't want to have these handcrafted priors, aka a hypothesis. Instead, we propose to condition the diffusion model via our image-to-brain encoder. During inference, we perturb the denoising process using the gradient of the brain encoder maximization objective, where gamma is a scale. We perturb the denoising process using the gradient of the brain encoder maximization objective. S in N are the set of voxels used for guidance. So this is S is a subset of N. So N is the entire flattened brain activation. So there's N total voxels and you flatten the whole thing, but N is probably huge. So they take a subset of that called S. And the reason they're taking the subset of that is because here they have a summation over all S. So obviously if they did a summation over all N, it would probably be extremely computationally intensive. So by taking a subset, they basically have a smaller summation here, which means they can actually do it in a reasonable amount of time. What is d omega here? This horseshoe is omega. Did they define that? Did they define d? They did not. What is d? Decoder. A learned image encoder and decoder. We utilize a pre-trained latent diffusion model with a learned image encoder and decoder. So the diffusion model, which is this little, this one here, doesn't operate in image space. It operates in latent space, right? So in order to perform diffusion, you need to take the image and turn it into a latent. So that's what's happening here. Now we finally kind of understand what's going on here, is that this encoder here is encoding the vector of brain activations, the of length b, and it's turning it into an image. But in order for that image to be used in this diffusion process, you need to encode or turn it into a latent diffusion model. So that's why you have this decoder. It goes from the image back to the latent. That doesn't make any sense. Wouldn't that be an encoder? A pre-trained latent diffusion model with a learned image encoder. The image encoder goes from the image to the latent. So why are they taking the image and using a decoder to turn it into this X? You can't go backwards through a decoder. This is just straight up wrong, right? The flow is this way, right? You feed an image into an encoder and get the latent. You feed the latent into a decoder and get the image. You can't feed an image into a decoder and get a latent. You can't go backwards through the decoder. That doesn't make any sense. I guess maybe the only way this makes sense is that they're showing you that where the gradient kind of goes. That's kind of what they care about. Yeah, okay. It's because the flow of information is these black lines. So they're not feeding the image into the decoder. What this is showing you is the gradient path. So the vector here, the embedding, the latent vector, is going into the decoder. It's producing an image. That image is going into the brain encoder and producing the brain activations. But then the gradient is the backward right? Is the back propagation with the chain rule is backward. So the image is not going from here to here. The image is going from here to here, but then the gradients are going back. Okay, so now this makes sense. All right. For the billionth time, it makes sense now. Okay, so here you have, we seek to maximize the average activation of S predicted by M theta. So E theta, I don't know why they didn't just write it like this. That seems like an easier way to write it. 1 minus alpha T, so probably a diffusion schedule. Yeah, so value of alpha interpolates between a normal distribution and P of X. The noise level varies by the time step T. Yeah, so alpha T is basically, in these diffusion models, because they work over a variety of time steps, usually what ends up happening is that the amount of noise that is added or removed is going to change, is going to be dependent on the actual time step itself. So that's what this alpha T is. So 1 minus alpha T times grad xt of this scale divided by the magnitude of S. So here, magnitude of S and then this summation over S means that they're just averaging over all these S. And S is a subset of N, which is basically just some subset of all the voxels inside your brain activations. You have your decoder, which is consuming the latent. It's producing an image. That image is being encoded with the brain encoder. And then you're summing over all of those to get the average activation. And then that average activation dude, what? We observe that the convergence using the current denoised xt is poor without changes to the guidance. This is because the current image latent is high noise. Yeah, this is like sometimes these diffusion papers are super confusing because they like swap, right? They say image, but it's not image, it's a latent, right? And here it's extra confusing because their brain encoder goes from image to brain activations. I would have almost made it go directly into the latent space of the diffusion model. I feel like that would have been cleaner, and you don't have to deal with this extra encoder decoder crap. We instead use a weighted reformulation with a Euler approximation of the final image. X hat 0, 1 over alpha. Alpha is just a fancy... Where is the equation for alpha? They don't even give you alpha. I guess this is supposed to be alpha t here. Xt minus 1 minus alpha t sigma t. Oh, okay, so this is now the noise here, this epsilon t or sigma t. I always get my sigmas and alpha and epsilons confused. Of course, the model that encodes the brain is going to be differentiable because you need to push gradients through it. I'm going to be honest, I don't really understand what's going on here. We perturb the denoising process using the gradient of the brain encoder maximization objective. So perturb the denoising process means that the amount of noise that they're adding, right, they're perturbing, they're disturbing, they're changing the noise based on the gradient of the brain encoder maximization objective. So the brain encoder, they're trying to use that to match this activation. The activation is not going to be, or the matching isn't going to be perfect, so there's going to be a gradient. The magnitude of that gradient is going to basically tell you how different your guess is to the actual thing. And depending on that, right, the magnitude of the gradient, so how bad is your guess, they're going to perturb the denoising process. Like, I feel like I can kind of see what they're saying here, but I just don't understand why they even did this. This seems like very random and confusing. I don't know. Drac, maybe you can help us. This is not a good showing right now, to be honest. I'm looking like a complete fool here. Mind's Eye consists of two pipelines, a high-level semantic pipeline where fMRI voxels are mapped to clip VIT image space. This seems much more obvious to me. Low-level perceptual pipeline where voxels are mapped to the image embedding space of a VAE. And this is what I felt like that first paper should have been doing, is just map these voxels, map those brain activations directly to the image embedding space where you're going to be able to do diffusion. Like, why map it why map it to the image space, which is what they're doing here, right? Why do this mapping to the image space here? I don't like the fact that this is an image. For the high-level pipeline, the submodules are an MLP projector and a diffusion prior. For the low-level pipeline, the submodules are an MLP projector and a CNN decoder. So you have two pipelines. Each pipeline has two submodules. So you have MLP projector, you have a diffusion prior, you have an MLP projector, and a CNN decoder. For both pipelines, we observed the training projector submodule with contrastive loss, and the second module with mean squared error loss gives the best performance. So the MSE loss is for the second part here. Oh. This is kind of cool. So you see here, they're actually taking, this is a different MLP than here, right? So here they're starting from the same fMRI voxel. So this and this is the same, but this MLP here is different than this MLP here, right? Notice how the output of this is different dimension than the output of this. So they take these here, they upsample it with a convnet into this here, right? This latent vector here, this 4x64 by 64. And then this is where the MSE loss is. The MSE loss is between this here, this vector that you get from this MLP, and then this convnet that upsamples, and then the latent vector for the stable diffusion image. So because this is locked here, right, it's like, where's the MSE loss here going to? It's not going into this encoder here, right, because this is locked. It's actually going into this upsampling and then this MLP here. So this MLP and this upsampling are trained to match the output of the image encoder here. So this is one training process here. You're training this stuff, and that is happening separate from this stuff here. This is a different training process. And then once you're done with both of these training processes, then you can take the fMRI, feed it into here, feed into this MLP, upsample that, get a latent, feed that into the decoder, which is also frozen, so you haven't pushed any gradients into here or here, and get this, what they call a low-level reconstruction. I would just call it an approximate version of the image, right? That's what I'm saying. I wouldn't use the word low-level here. I would just say this is just a approximate version of the image. It's just because if this was way better, you would probably get way better images, but because it's not super good, it's just an MLP with an upsampling convnet, you're not necessarily getting very high quality images. And then because it's not a very high quality image, they want to basically make it look nicer, and in order to make it nicer, that's where they use this extra diffusion process, where rather than starting from random Gaussian noise, they start from this low-level reconstruction. High-level semantic pipeline is the core of Mind's Eye as it maps voxels to clip image space to be fed through pre-trained image generation models. We refer to it as a high-level pipeline because clip embeddings are inherently more semantic than perceptual, since clip image encoders were trained to maximize similarity with text captions. Low-level features like color and object location are not typically preserved in these captions. Okay. Yeah, so finally they explain that high level and low level are like kind of hand wavy concepts, to be honest. I don't like this because up until this point I was like very confused about this like high level, low level, because in my head I was thinking kind of this image of like high level, low level in convnets, and I thought they were kind of more specific, like they were maybe doing some kind of hierarchy thing, but it's not. It's like they're interpreting this as low level reconstruction and this as kind of a high level reconstruction, but it's not. It's all hand wavy. The MLP backbone maps flattened voxels, so they're flattening the voxels. Did these guys also flatten the voxels? I think these guys also flatten the voxels. Yeah, so brain activation fMRI represented as a vector, and notice how this vector isn't two-dimensional or even three-dimensional. It's not n by n by n, it's n, so they're flattening the brain activation, and the same thing is happening here, right? They're flattening it to an intermediate space of 257 by 768, corresponding to the last hidden layer of the clip VIT L14. The backbone consists of a linear layer, so they flatten it to this because that's the exact size of the clip VIT. So clip VIT is a vision transformer that is going to convert an image into a latent vector, and they need to do a contrastive loss, right? So they need to basically get a distance metric between this and this, which is going to be the cosine similarity, probably. But in order for that to be possible, they need to be able to basically have the same exact dimension. So that's why their MLP backbone here and projector are the same dimension as the clip. The backbone consists of a linear layer followed by 4 residual blocks and the final linear projector. The embedding, so I still feel like they could have done like a transformer or something a little bit more modern. Like these just seem very primitive. The embeddings from the backbone are fed into an MLP projector and a diffusion prior in parallel. The whole model is trained end-to-end with the prior getting an MSC loss and the projector getting a bidirectional clip loss. The projector outputs can be used for retrieval tasks, and the diffusion prior outputs can be used by generative models to reconstruct images. So they're saying they're doing both of these at the same time. The contrastive loss and this MSE loss. It's not like they're doing one and then the other. They're basically, I guess, alternating them, or the loss that they use is a weighted combination of both of these losses. A weighted combination of a contrastive loss and an MSE loss, though, that's going to be, you're going to have, like, the weights for those are going to be a little bit weird to get. Contrastive learning is an effective method for learning representation across modalities by maximizing cosine similarity for positive pairs while minimizing similarity for negative pairs. Okay, so they are going to be using positive and negative pairs. The pairs there refer to the fact that this image corresponds to this fMRI voxel or voxels, but you can also say, okay, well, this image corresponds to these voxels, but here's a bunch of random unrelated images which I just sample from Layon 5B, and those are going to be my negative examples. So you have positive examples and negative examples. CLIP is a multimodal contrastive model that maps images and text captions to a shared embedding space. Mind's Eye is trained to introduce fMRI as an additional modality to the embedding space. This sentence right here is literally this entire paper. Is basically, they're just using the power of the pre-trained CLIP model. They don't fuck with the CLIP image encoder. They don't, as done with locked image tuning. This is a paper that we read where they alternate freezing the text encoder and the image encoder, and they try different variations of freezing the text encoder, freezing the image encoder, and so on. We use the CLIP loss as our contrastive objective. This loss is bidirectional and helps improve both the image and the brain retrieval. I don't think they're pushing gradients with that. I don't know what they mean by bidirectional. There's no gradients going into this image embedding, so it's not like this is changing at all. Recent work has explored novel data augmentation techniques that offer several benefits like improving performance, increasing robustness, and reducing training data requirements. Mix-up is one technique which trains models on synthetic data created through convex combinations of two data point label pairs. So this is the mix-up. We found a picture of that actually looks quite nice. Where is it? Mix-up. Yeah. So this is the combination of data known as mix-up. So a little bit of data augmentation there. MixCo, an extension of mix-up that uses the info NCE loss and show that MixCo improves classification performance in a semi-supervised setting. Based on the same principle, we modify the bidirectional clip loss to use MixCo, while Kim et al. Observed that MixCo gives the largest performance benefit for smaller models. We observed that it also helps large models in low data regimes. Okay, so this MixCo that they're using, in the original MixCo paper, they were using it to improve the performance of smaller models, but here they're using a very large MLP and a very small amount of data. So this is kind of the large models in low data regime is like overfitting danger zone. So that's why they wanted to use it. To combine MixCo with a clip loss, we mix voxels using a factor lambda sampled from the beta distribution with alpha equals beta equals 0.5. Okay, so choosing these parameters here. So much like in the normal distribution, you have to pick your mean and your variance, and with those two numbers, you can define the shape of the Gaussian or the normal distribution. The same thing with a beta distribution, you have to pick these two numbers, alpha and beta, and that'll define the shape of the beta distribution, which they then use to get this lambda number here. xi and yi represent the ith fMRI sample and image. So xi is the fMRI sample, and then yi is the image. So usually in a machine learning model, x is the input and then y is the target. So here they're letting you know that the image is the fMRI is the input, and then the image is the target. So the image is lambda I times xi, and then 1 minus lambda I times xki. So this is the cut mix, right? So this is what we were looking at this, right? It's like the image that you actually end up feeding is basically a combination of two existing images with whatever this is, 30%, and then 1 minus 30%, which is 70%. So that's what this is here, right? Lambda time, and then 1 minus lambda. Pi star equals f x mix I ki. What is pi star? P star, P, and T are L2 normalized. Okay, so you have k possible mixings, right? You're mixing together two things, or two x's, which are different fMRI samples. You mix them together, and the output should basically also be the mix, should also correspond to the mix of those two outputs. Ti is the output of the clip image model when you give it yi. So the clip image model here is just the clip encoder. So this is yi, and then this is ti, right? So ti is the actual embedding vector that comes out of the clip image encoder, which takes in the image y. Okay, so the clip loss with the mix co is defined as this giant, this is like the grossest equation we've seen all week. It's a combination of the clip loss with this mix co loss. We term this loss by mix co. Tau is a temperature hyperparameter, and n is the batch size. So you're summing over the entire batch. You have different lambdas here. There's a lambda I, and then there's also a lambda j. I guess there's two different summations here. This one's over I, this one's over j. This one's over L. So you also have a lambda L here. Oh, shit. You see this here is the cosine similarity. You see this dot product, right? Pi and T should be the same, right? T is what's coming out of the clip image embedding or encoder, and then P is what's coming out of your brain, what do they call it? The brain encoder, whatever, right? So you want this value here, this cosine similarity, this dot product, you want those vectors to be as similar as possible, right? Let's see if we can get, yeah, you want the vector that corresponds to the fMRI to be very similar to the vector that corresponds to the image. So you want this part here that's coming out of the clip image encoder, this clip image here, which I think they call T, you want that to be basically the same as this here, which they're calling P, right? So P and T should be the same, which is what you're seeing here. This loss is basically just a bunch of dot products between P vectors and T vectors. See that? It's basically just you're trying to make these dot products as small as possible. Recent works have shown that stopping mix-up augmentation after a certain number of epochs leads to better classification performance. Okay, so apparently mix-up augmentation, you don't want to use it the whole time. You want to basically use it only at the beginning. We stop using mix-up and switch from a hard contrastive loss to a soft contrastive loss 1/3 of the way through training. Okay, so another secret hyperparameter here is that there's some schedule to this, and it's basically 1/3 of the way. I don't know where they got that 1/3. They probably just got it from the mix-up paper, but, you know, gotta love arbitrary random hyperparameters like that. our soft contrastive loss is inspired by knowledge distillation, where the authors argue that the softmax probability distribution produced by a powerful teacher model acts as a better teaching signal than hard labels. Okay, so what do they mean by this? So whenever you're doing a classification model, right, the output of your classification model is, these all suck. Let's do classification model neural net. See if I can find a good pick here. This one? I'm like really trying to find a specific pick, but I can't find it. Yeah, maybe this is a good pick. So the output of your classification model is not going to be one number. It's going to basically be what's called the logits, which then get put through a softmax and turned into confidences, right? And the confidence kind of, you could think of, let's say this is the logits, and then this is after this, actually, no, never mind. This is the logits after they have been fed through the softmax, and the softmax is going to turn them into probabilities between 0 and 1. So you can say, okay, this is 0.6% chance of cat, 0.1% chance of dog, 0.3% chance of chair, right? If you had some classification between dog, cat, and chair. So what they're saying here is that if you have a teacher model, basically a neural network that has already been trained on this task, you feed the image through the teacher network, and the teacher network is going to give you 0.1, 0.6, 0.3. That is a much more useful label to train a network than if you just had a hard label. So by a hard label, what do they mean? Well, a hard label would be 0, 1, 0, right? The hard label doesn't have specific numbers for basically any of the other classes. It only tells you that this image corresponds to one specific class, which is cat. So if you were to train a student on hard labels, which is kind of basically the original labels, everything, you're basically training it on a vector. You're training it to match a vector that has 0, 1, and then 0. But if you're training it with a teacher model, you're training it with a more kind of nuance. There's more signal here, right? The teacher model is giving you a probability over all of the classes, which means that you can leverage the fact that, hey, this picture is a 0.1 dog versus a 0.05 dog, right? So there's more signal in the softmax probability distribution produced by a teacher model than there is in a hard label, which makes sense. To generate the soft labels, we take the dot product of clip image embeddings in a batch with themselves. The loss with bidirectional component omitted for brevity is calculated between clip and brain clip matrices. Okay, so what they're using here is the loss that they're using is basically the cosine similarity between these two vectors here, right? They want the vector that comes out of the clip image encoder and then the vector that comes out of the fMRI encoder to basically be the same. They want that dot product to be high for related things and low for unrelated things. So what they're saying here is that since they're using a bunch of positive examples and negative examples, right, they could just say, hey, this is positive examples, aka 1, right, hard labels, 1 for positive examples, 0 for negative examples, or they can say, okay, well, actually, let's take the dot product of these clip image embeddings, and that's going to have a little bit more signal because you're going to be able to, it's not just going to be basically a bunch of zeros and ones, it's going to be like more nuanced numbers. So that's why they're the soft clip here versus hard clip. So soft is the 0.1, 0.6, 0.3. Hard would be 0, 1, 0. And that's what this loss is. So you see here how now in this loss, they're multiplying the PI, which is what's coming out of the fMRI embedding, with the TI, which is what's coming out of the clip. So this is a dot product between the fMRI embedding and the clip embedding. But you see here how now they have TI times TI here. So the dot product between the different embeddings that come out of the clip itself. So it's not just pi.ti, it's also ti.ti. Although here it's like i's and j's representing the different indices of the batch size, where n is the batch size. right? So if there's 10 images in the batch, maybe you have 5 negative examples, 5 positive examples, and so on. We use a diffusion model to align the outputs of a contrastive learning model where a diffusion prior was used to map the clip text embeddings to clip image space before using an unclipped decoder to reconstruct images. Inspired by DALL-E 2, you have a diffusion model which maps clip text embeddings to clip image embeddings. What? We modify an open source implementation of DALL-E 2 diffusion prior available on GitHub. And then the total final loss is basically the combination of this bimix co or soft clip, which they switch a third of the way through. So at the beginning, you're using this bimix co loss, and then a third of the way they switch into this soft clip loss, and then they have this what they're calling the diffusion prior loss multiplied by this weight here, alpha, which they just decide as 0.3. We switch from big mix code to soft clip after 1/3. All of our models are trained on a single A100 GPU for 240 epochs with a batch size of 32. So good to know that they're not using a huge amount of GPUs. This is definitely just a standard research kind of budget here. Diffusion prior is critical for reconstruction because contrastive learning only incentivizes the clip fMRI embeddings to match the vector direction of the associated clip image embeddings. This generates disjointed embeddings as observed by Ramesh. To rectify this issue, the diffusion prior learns the distribution of clip image embeddings conditioned on clip fMRI embeddings. Because contrastive learning only incentivizes the clip fMRI embeddings to match the vector direction of the associated clip image embeddings. Yeah, okay, so I agree with that. This generates disjointed embeddings. To rectify this issue, the diffusion prior learns a distribution of clip image embeddings conditioned on clip fMRI embeddings. UMAP 22 plots of the disjointed next to aligned in appendix A4. Let's go to that. UMAP comparison. Okay, so there's aligned and unaligned. Where is it? Where's the UMAP? As depicted in Figure 9. Where's Figure 9? Figure 5, Figure 6, Figure 7, Figure 8, Figure 9. Here you go. UMAP. UMAP is kind of like Teasney. It's like a dimensionality reduction technique usually used to create these kind of plots. So what they're showing you here is that you have the clip image embeddings. You have the MLP backbone, which I think is the embeddings that are coming out of the fMRI. You see here how they're obviously in two different kind of groups there, clusters. MLP projector latents, green, and mind's eye diffusion prior latents, red. UMAP were estimated from 1,000 random samples. Clip image latents correspond to the last hidden layer of VITL14. Euclidean distance between the given mind-eye embedding space and clip image space is shown is lowest for the diffusion prior, suggesting that the diffusion prior helps to align the two embedding spaces. I mean, this is hard to tell, but supposedly this is unaligned and this is aligned. Kinda. Right, here they're showing you the distance here, 0.4 versus 0.1 versus 5.8. I don't know, maybe we need to read that paper where they use the diffusion prior. That's what this part here is. right? So it looks like this diffusion here is actually diffusing directly into clip embeddings. And that clip embedding should match this clip embedding with an MSE loss. But they also are pushing gradients into this MLP backbone. So there's two kind of paths for the gradients to flow. One of them is here through the contrastive loss, right, which is the cosine similarity, into this MLP projector, into the MLP backbone. Another path for the gradient is through this MSE loss into here, into this diffusion prior, which is conditioned on the MLP backbone. So then it goes into the MLP backbone. So the MLP projector is only receiving gradient updates from this contrastive loss, versus the MLP backbone is receiving gradient updates from not just the contrastive loss, but also this MSE loss that's going on through here. There's a trade-off between reconstruction and retrieval objectives, and a model cannot effectively learn a single embedded space that does well on both. So you have to basically reconstruction versus retrieval, there's a trade-off. Why would there be a trade-off between reconstruction and retrieval? Wouldn't a embedding space that's similar to the clip embedding space be the best for both? I need to drink more coffee. The low-level pipeline maps voxels to the embedding space of Stable Diffusion's VAE. The output of this pipeline can be fed into the VAE decoder to produce blurry image reconstructions that lack high-level semantic content but exhibit state-of-the-art low-level image metrics. We use image-to-image to improve our final image reconstructions in terms of low-level metrics with minimal impairment to high-level metrics, such that we start the diffusion process from the noised encodings of our blurry reconstructions rather than pure noise. Yeah, so what they're referring to here is this part of the pipeline here, right, where there's a diffusion happening here, and then there's diffusion happening here. And this diffusion, which they're calling image-to-image, is basically just rather than starting from Gaussian noise like most diffusion models do, right? Diffusion model. Right? Rather than starting from this, maybe not that maybe that's not the best example. I kind of like that one picture of the cat. Yeah, here you go. Rather than starting from this, right? This complete basically noise and then going back to the actual image, they're going to start from the kind of blurry image that comes out of this Stable Diffusion VAE, and that's what they're going to use, and they're going to basically do however many steps of diffusion here conditioned on this clip fMRI embedding vector here. And this is frozen, and how can they keep this frozen? They can keep this frozen because this vector here should ideally be as very similar to the clip vectors that come out of this frozen model here. And this model was probably pre-trained on clip vectors. So that's probably why it doesn't matter and it works quite well. The MLP backbone for a low-level pipeline follows the same architecture, except that the final outputs are of size 16 by 16 by 64. They're upsampled to 64 by 64 by 4 by a CNN upsampler. An MLP projector projects the backbone outputs to 512-dimensional space, where an auxiliary contrastive loss is applied. For more information on the low-level pipeline, see Appendix A2. All right. So we've made it through the implementation section for both these papers, but we're obviously beat up and bloody at this point because I think we've kind of sufficiently confused ourselves. But now we're in the results section, so we can take a breath of fresh air because this part is easy. So let's kind of compare what they're comparing to. So they're going to be comparing to the natural scenes dataset. I think that's the same one that we have here, the natural scenes dataset. So both of these are using the same dataset to compare to, which makes sense. This seems to be a public fMRI dataset containing responses of human participants passively viewing natural scenes from MS COCO. Okay, so people inside some MRI machine that are getting shown MS COCO images, and then they measure the brain responses. Standardized train test splits. We average across 3 same image repetitions and not the training set, similar to Takashi whatever. Okay. That's exactly what happens here, I'm guessing. All images are from the MS COCO. The beta weights or activations computed using GLM single and further normalize each voxel to a mean of 0 and a standard deviation of 1. So they're normalizing the activations of the blood flow in each voxel, right? That's what each voxel is doing, is it's basically getting some estimate of the amount of blood going through that region in your brain, which is why you have to actually sit very still in an MRI machine, is because if you move, now you've shifted, right? The voxels aren't moving, but your head is moving, so you have to be very still. The 9,000 unique images. We utilize Stable Diffusion 2.1 base, so all these different pre-trained models that they have are Stable Diffusion. We use multi-step second-order DPM solver. This is the noise sampling. There's a bunch of different ones of these with 50 steps of diffusion. We set hyperparameter n equals to 130. Images are resized for the brain encoder. Null prompt is used as an input prompt. Thus, the diffusion performs unconditional generation without brain guidance. They use the same vision transformer. Do they use the same vision transformer? I think these guys use VIT huge. Yeah, so this paper is using VIT-L, which is bigger than this paper, which uses VIT, or is VIT-L bigger than VIT-B? VIT-B versus VIT-L. VIT-L and VIT-B. The L model is significantly larger, hence it performs better, and it's also slower. Okay, so that's important to understand, is that this paper is using a much bigger image encoder here, VIT-L, versus this paper uses a VIT-B. Although this one seems like it used VIT-L as well. I don't know. Place voxel, face voxel, food voxel, body voxel, word voxel. Concern Doge, the Discord invitation doesn't work. Yeah, sorry. I just keep fucking with these Discord channels and it keeps messing up this Discord invitation link, but I appreciate you mentioning that. I will go ahead and change it. Okay, so I mean, these kind of seem categorically correct. This does seem like pictures of food. This does seem like pictures of faces and pictures of bodies and pictures of places, so that kind of seems fine. We asked human evaluators to select which of the 2 image groups scored higher. $12 an hour to get your brain scanned. Here is the actual regions of the brain. So you can actually get an idea for the resolution of the voxels here, right? So just like if you see the tiniest dot you can see, that's kind of going to be 1 voxel probably. All right, so let's hope that they actually use the same metrics here. Given a brain sample and an image, this is the retrieval. So here you go, select the highest clip cosine similarity. So within Leon 5B, which is the image that is the same. Top 1 versus top 5 basically means, are you picking the image with the highest cosine similarity, or are you picking 5 and then seeing if it's within the top 5? Top 5 is easier than top 1. Having a good top 1 score is way harder than having a good top 5 score. Image. Here are the scores. High level, low level, retrieval. This is not the same metrics that they're using here. Unfortunately, these are different. They don't even compare. NSD. So we can't actually compare this paper versus this paper. They're using different tables here. Brain Dive generates images with higher semantic specificity than the top 1% of natural images for each brain region. Here they're comparing to Big GAN. I don't exactly know what I'm supposed to be seeing here. Results for face selective regions of interest. So I guess they constrained to specifically face ROIs. ROI is not the mask RCNN concept here. I think ROI refers to the region in the brain. Selected using betas, average within each ROI. Betas, from what I can glean, is basically just the activations. Are ranked according to their official predicted ROI activity means. Brain Dive are ranked using our predictive ROI from 500 images. So I guess what they're showing you here is that this red shows that you can still get activations for non-face related things inside this. This one's kind of cool. It's the face of a dog, right? So the face of a dog maps in your brain to the face selective ROI, which I think makes a ton of sense, right? It kind of makes sense that your brain would activate in the same way to the face of a dog than the face of a human. Suggests little differentiation between ROIs within the place, word, and body selective networks. We also compare results against the top images for FFA and OFA for neurogen. We observe that both diffusion-generated and NSD images have a very high face content in FFA, whereas neurogen has higher animal face content. OFA versus FFA. Comparisons are done with within GT and generated images respectively. Which group of images has more X? So basically they want all of them to be faces. We observed text selectivity and face selectivity in S5. By avoiding the use of fixed categories, Brain Dive images are more diverse than those of neurogen. This trend of face in animals appears at threshold. So I think kind of what they're trying to say here is that, I don't know this work neurogen, I haven't read this paper, but my assumption is that maybe neurogen conditioned on or used the prior of like knowing what these regions are. So like in the neurogen paper, I'm sure they maybe used some additional signal that said, hey, this region corresponds to faces, this region corresponds to food. But in this paper, they're not doing that at all, right? They just kind of treat the activations as just this giant set of voxels. So maybe that's what they're trying to say here. To quantify, we perform human studies, acts to compare. This difference is on average, indicating that our approach is able to highlight subtle differences in semantic selectivity across regions. First, perform clustering on normalized per voxel encoder weights using VMF clustering. We find consistent cosine differences between the cluster centers in the food selective ROI as well as the occipital place area. We observe a relatively consistent anterior-posterior split of the OPA. While the clusters within the food vary more anatomically, each subject appears to have more medial and more lateral clusters. This is all just like brain terminology here. I don't know exactly. I'm guessing anterior posterior is like more forward of the brain and then more to the back of the brain and then more to the center of the brain, which is medial, and then lateral is to the side of the brain. So it's kind of like boats where it's like starboard and then there's like specific words for like all the different cardinal directions in a boat, and I assume that there's basically the equivalent here for brains where there's different cardinal directions in the brain and they have like their own jargon for which region is which. Food cluster generally lacks vegetables, mostly consistent of bread-like foods. Cluster 1 is dominated by indoor scenes. More human studies. More angular geometric, more indoor scenes, less natural. So basically, it sounds like they're realizing that these different regions are actually, they're a little bit more nuanced, right? Like it's not just food versus faces. It's like all these like subcategories, right? Like vegetable and fruit. Like probably your brain thinks of a tree more similar to a vegetable than it does the tree to a steak. Maybe the steak is more like the face. Like, so here it seems like there's a bunch of like cake looking things. Like these are what they call bread-like foods. More visible high color saturation foods. Which cluster is more? Difference between OPA clusters. Visualize top 10. While both consist of scene images, the first clusters have more indoor scenes, the second has more outdoor scenes. So here, I guess you have more outdoor, and then here this is more indoor. Okay. All right, so that's the results section. It seems like this paper was really kind of trying to dive deep into this. What do the clusters represent, right? Obviously, the people who wrote this paper are in the brain world, so like they're not necessarily interested in some random arbitrary benchmark. They're actually interested in like, what do these different things mean semantically in your brain? Like what are these clusters? Within these clusters, do you have like a difference between different faces, different animal faces versus human faces, indoor scenes versus outdoor scenes? So most of this results section is basically analyzing those clusters and the semantic meaning of those clusters. Let's look at what this paper has to say. So this paper, these are more standard metrics here. So you have pixel correlation, you have AlexNet. Sometimes this is the distance of an AlexNet. So you take an AlexNet, you feed the image through an AlexNet, and then you take the very last embedding, right? And then you compare it. So it's a way to compare two images is the distance in AlexNet space. Inception score is also very similar, except it's using an inception net and the distance in the embedding space of an inception net. PixCore, pixel-wise correlation between ground truth and reconstructions, SSIM, structural similarity index metric, EfficientNet, EF, SuaveResNet refer to average correlation distance. All other metrics refer to two-way identification. Missing values are from papers not reporting all metrics or metrics being non-applicable. Okay, so this paper is all about retrieval and generating a specific image. So they're less interested in this kind of clustering and like understanding what the semantic concepts of each cluster are, and they're more interested in just trying to produce the best possible image conditioned on the brain activation. So what they want is they basically just want their reconstructed image to be as close as possible to the real image. And here are basically other approaches that are also doing that. So these all kind of basically look the same to me. Honestly, it's like train, person surfing, baseball. Like, I don't know. It seems like they're kind of, all these papers are really competitive. They're all pretty much on par. Here they're trying to say that they're better, but I mean, it's like barely better, right? Like 97, 96, 91, 94. These results are very, very close. Maybe this one's a little bit better here, 21% to 93%. Okay, so they do some ablation studies. What do they ablate? Probably just different sizes of ResNets and transformers. So here they have the MLP, right? So they have this multi-layer perceptron up here, MLP backbone, and then they have another MLP here, right? So two different MLPs here, but one of the ablations seems to be the size of the MLP. So they have here different sizes of that MLP, and then whether or not it has residual connections. So big model, small model, big model. Obviously, seems like the big model does the best. But interestingly, the brain retrieval model is capped out. You see that? So there's more of a difference between more of a relationship between size and performance on image retrieval than there is between size and performance on brain retrieval. Training strategies. Okay, yeah, this was the paper that had all these like complicated like losses here. So they had the bimix co, which they switch with the soft clip a third of the way through. So here they're trying probably different schedules. And you see that it does kind of, you do want this soft clip loss, right? You look at the difference between this one 0.3 and this one 0.8. So it does seem like this soft clip versus not soft clip makes a big difference. This is fucking weird though. The fact that the brain retrieval and the image retrieval almost seem to be like there's a different relationship between these hyperparameters here and then the performance on these two separate things. That's a little weird. Right? You would have thought that they would have been basically the same, but they're not. Okay, so without that diffusion prior that they were using, which was pushing additional gradients into that brain encoder, the model does poorly. And I think that's another thing to realize is that sometimes when you read these papers, it kind of feels like this giant collection of crap, and it's like, and it's confusing, and it feels like how did they come up with this weird ordering of like weird specific little things that all feed into one another? And I think that the reality is that the way that these projects work, it's like they probably tried a much simpler version of this and then it didn't work, and then they added an extra thing, and then that didn't work, and then they added this thing over here, and then now suddenly it works. So it's not that they sat down in day zero and like came up with this like weird like MSE loss and contrastive loss, both pushing gradients into this MLP backbone. It's that originally they would have very much liked it just to be like this, right? They would have very much liked it if all they had was just a contrastive loss and that was it. But it didn't work, so they added this extra thing here, and then it kind of worked. So these kind of like systems and the way that they like all these different parts of the system, like they're basically the result of a iterative process that usually adds complexity over time, right? The complexity gets higher as you go deeper into this research project and you try to find more and more things that you can add, more auxiliary losses, more additional modules that'll give you that performance that you want and that result that you want. So yeah, it's a lot of different moving pieces, but it's not because they designed it that way originally. It's just because that they just kept trying to do shit, and then they figured out that, okay, well, we need to add this extra thing here in order for it to perform better. All right, related work section here. And then conclusion. All right, let's read the conclusions, and then we'll be done here. Conclusion. We present Mind's Eye, a novel mental decoding approach that achieves state-of-the-art reconstructions of natural scenes presented to humans in the MRI machine. These reconstructions retain semantic meaning and perceptual similarity to the original images due to the use of a combined high-level and low-level pipeline. I didn't like this. I didn't like this like weird hand-wavy kind of justification that they did where one thing is more high-level and one thing is more low-level. The novel use of specialized submodules for contrastive-based retrieval and diffusion-based reconstruction allow Mind's Eye to learn mappings for both in tasks in parallel. Mind's Eye can select the ground truth image out of a set of nearly 1,000 possible images with 90% accuracy, suggesting fine-grained image-specific signal contained in the brain embeddings. Mind's Eye retrieval can also be used when the original image is unknown by querying large databases such as Layon 5B. The diffusion prior submodule allows for accurate translation of brain embeddings into pre-trained clip space such that any model that accepts clip embeddings can be provided with clip fMRI embeddings without fine-tuning. Yeah, this is huge, right? Because it means that if someone trained a diffusion model or any kind of generative AI model that accepts clip embeddings, you can just now give it these clip fMRI embeddings. Mind's Eye reconstructions will continue to improve as newer, more powerful image generation models are released. Privacy concerns, of course, if you can read someone's memory, it's going to be a little bit creepy because people are going to be able to know what you're thinking. Open developed through volunteer contributions into the MedArc Discord server. Source code is accessible via public GitHub. We continue to extend global invitation to the Mind Reading Lab project. Okay, so this is where the Eleuther AI and Stability AI comes from. We saw those at the beginning of the paper. Oh, I didn't realize Jeremy Howard is part of the MedArc community. That's kind of cool. And then this is what all the people actually did. Cool. What about this one? We showed that Brain Dive generates diverse and realistic images that can probe human visual pathway. This approach relies on an existing large data set of natural images paired with brain recordings, in that the evaluation of synthesized images is necessarily qualitative. Important to validate whether our generated images and candidate features derived from these images indeed maximize responses. Future work should involve the collection of human fMRI recordings using both our synthesized images and more focused stimuli. We introduced a novel method for guiding diffusion models using brain activations, enabling us to leverage generative models trained on internet-scale image datasets for data-driven explorations of the brain. This allows us to better characterize fine-grained preferences across the visual system. We demonstrate that Brain Dive can accurately capture the semantic selectivity of existing characterized regions. We further show that Brain Dive can capture subtle differences between ROIs within the face selective network, and we identify and highlight fine-grained subdivisions within existing food and place ROIs, differing in their selectivity for mid-level image features and semantic scene content. We validate our conclusions with extensive studies. Okay. It's been a long journey to get to the end of these two papers. I'm not going to lie, I'm a little bit confused. Probably my brain mixed up a bunch of concepts from both these papers, but let's see if we can try to summarize these papers and then end it there. So today we read two different papers. We read two fMRI papers that use diffusion models, one coming out of Carnegie Mellon and then another one coming out of a more open source ensemble of researchers. Both papers use fMRI, which is basically a functional magnetic resonant imaging, where you get a proxy for neural activity in the brain by looking at the blood oxygenation within a specific voxel of 3D space, right? So you basically have these 3D voxels all over in some specific part of your brain. You measure the blood flow through those, and you flatten them, and you get this very long vector, which represents the activity of your brain. In this paper, so in the Carnegie Mellon paper, the technique that they were using was basically they trained an additional brain encoder and decoder, and then they use basically that latent vector here to condition a denoising diffusion model here, right? A diffusion model that works in latent space. Latent diffusion model, pre-trained latent diffusion model, which is stable diffusion 2.1, right? So that's basically what they're doing. Here they're using CLIP. Here they're also using CLIP. So both of these papers use CLIP as kind of like the fundamental embedding space, if you want to think of it that way, right? So many papers in computer vision are basically building on the fact that the CLIP embedding space is really good, and it has like a very good notion of semantic concepts. And in both of these papers, they do not push gradients into CLIP, right? Here they're freezing CLIP, they're only using it to get these image embeddings, and the same thing here, they're not pushing any gradients into CLIP. So CLIP is sacred, you do not push gradients into CLIP. What else? I think in this paper, I don't know if they did anything fancy, to be honest. It's pretty oh yeah, this was the paper where they did this like weird, yeah, this where's the sentence? There's a sentence here. Yeah, we perturb the denoising process using the gradient of the brain encoder maximization objective. I didn't really fully understand why they were doing this, but somehow that results in a better situation. In this paper, the weird thing that I didn't like was this double loss thing here, where they have the loss coming in at different levels of their fMRI encoder, right? They use an MLP here, backbone, and then MLP projector, and then they have losses coming in through here, and then losses coming in through here. I thought that was a little confusing. And then I didn't like the fact that they separated it out, right? Like, to me, they should have, if this paper was perfect, it would have basically just been a contrastive loss to train this decoder, and then this decoder, you could have just generated images directly from this embedding, right? But obviously they tried that and it didn't work, so then they added this extra diffusion stuff, and then they added this extra part here where they basically use the stable diffusion VAE to generate a kind of blurry version of the image, and then use that as the initialization for a second diffusion process where they actually remove more noise conditioned on this CLIP vector. So I didn't like this kind of like two-part pipeline. I think it's a little bit confusing, but of course they probably just had to do it because it didn't work in the simple implementation that they originally wanted. I think this is actually the cleaner way to do it, much simpler. Except for this weird thing that they're doing here with the perturbation. But yeah, that's it. Both of them kind of show results. The results seem kind of about the same, to be honest. This paper focused more on comparing to other existing works and showing that the generated stuff is, or not this paper here, was trying to compare to existing works and then show that the images that they generate are more similar to the image that was shown to the person in the MRI machine. This paper here kind of focused more on investigating what the actual clusters within the brain are, right? Like difference between faces, difference between foods, and so on. And that's pretty much it, guys. I'm just going to be honest. I felt stupid reading these papers, you know. I thought I understood diffusion models and CLIP, but, you know, these guys showed me that there's still more to learn. So sorry if some of that was confusing. Thank you, Drac and Fang Yi and Jonathan, for the questions. Drac especially. it seems like Drac is works in this field of brain imaging analysis. I don't know what the field is called, but thank you for the comments and questions and concerns. And that's it. Tomorrow, I think we have a paper on a hierarchical vision transformer. So that one should be a little bit easier. But try something a little new here. You know, I like stepping into different areas of research that I don't really have a background in, but you know, that's how you get smarter, is you try to understand and learn about papers that you don't really understand. So thanks for listening. Hope you guys have a great Wednesday, and see you guys later.